So now you're going to have a real scientist. It's a great pleasure to introduce Dr. Professor Paula Cohen. She came to us from England, as all the good things come from England. And she actually had her PhD in 1990s in reproductive endocrinology. Then she was a postdoc at Albert Einstein College of Medicine, where she was specifically studying reproductive biology and genetics. And subsequently, she focused on genetics, and she has been done tremendous work. She has a couple front-page journals, which for each of us, it's a holy grail of having your pictures on the Nature or some other major journal, of her work in the control of meiosis. Meiosis, this is how the cells divide, and she's going to be talking to you about this. She works at the veterinary school, although she's not a veterinarian, but as long as I've heard, Dr. Travis, who is a veterinarian, doesn't hold it against her. And she actually is now an associate professor, has been just promoted to be an associate professor of genetics. So welcome.
Thank you. It really is an honor to be here and to you see the clinical face of the work that we do every day, many hours a day, 7 days a week. We live it and breathe it. So it's nice to be here and see really why we're doing it. So what I'm going to do is sort of tell you a little bit about what our interests are in the lab and how they relate to the clinical syndromes that we're discussing here today, and other clinical syndromes, and really to try and bridge that gap that Dr. Schlegel was talking about between the basic scientist and the clinic. As Dr. Paduch mentioned, my lab is interested in this process called meiosis. It's the process that takes a cell with 46 chromosomes and makes 4 cells often containing 23 chromosomes each. And it really is the principal basis for our being here. It's the basic mechanism by which all sexually reproducing organisms propagate the species. And so it's quite obvious that it should be a very critically regulated event. So the defining stage of meiosis, and this is just a little cartoon that you might all remember from your biology textbooks about what meiosis is, but the critical defining stage of meiosis is this stage that we call prophase 1. And it's in this stage that the 46 chromosomes, the 23 pairs, one maternal, one paternal chromosome, have to find each other in this sort of chromosome dance. We really know very little about how chromosomes do that, how chromosome 1 finds its partner chromosome 1 and chromosome 2 and so forth. And that in itself could be the subject of many, many textbooks. This is the first event, how chromosomes find each other. And then the second almost miraculous event that takes place is these chromosomes pair in some way, in some physical way. So they exchange genetic information, DNA, to become intertwined. And now we learn from the shows on the Discovery Channel and from our biology textbooks that the reason that chromosomes intertwine in this way and recombine their DNA is to produce diversity amongst the species. That's rubbish. It's not. It's really just a very, very beneficial side effect. The only reason that we break our DNA and reform DNA across our maternal and paternal chromosomes is to keep those chromosomes together. So the best way to glue things together is to break them and recombine them. And so this stage of prophase 1, this stage at the top here where chromosomes are finding each other and pairing physically, is absolutely crucial to keep those chromosomes together and then to allow them to separate equally. So you can imagine all the different scenarios in which these things could go wrong. It's actually a miracle it ever goes right. So this is a stage that I'm interested in and my lab has been interested in for some time. We can break this stage down into substages, and these substages are defined according to how the chromosomes look at each of those stages. So prophase 1 is divided into 5 substages, leptine, zygotine, and pachytine being the first 3. And it's during these 3 stages that these chromosomes... Ooh, I just went echoey. It's during these 3 stages that these chromosomes have to find each other and undergo this physical breaking of DNA and recombination across maternal and paternal chromosomes. So I'm showing you here a cartoon of how these events happen. They happen primarily by the formation of this protein scaffold that we call the synaptonemal complex. And this scaffold aligns each chromosome. Okay, sorry. This protein scaffold starts to form early in prophase 1 and forms a scaffold along each chromosome. And then, if you imagine the function of a zipper, a central component of this synaptonemal complex then comes along and zippers the chromosomes together. So the first pairing, or what we call synapsis of these chromosomes, is a protein-driven event. So a scaffold comes in and glues these chromosomes together. Why does this happen? Well, what happens during this process of synapsis is that the DNA breaking and reforming events are taking place, the events of recombination. And so this protein scaffold basically resides throughout prophase 1 to act as this gluing mechanism to keep those chromosomes together so that recombination can take place between maternal and paternal chromosomes. So since this is so essential, I'll just go on and show you some of the images first, sorry. So this, on the right-hand panels, you've got the cartoon. On the left-hand panel, you actually have live cells or fixed cells that are doing this. And in red, you see the beginning of this synaptonemal complex protein that forms a scaffold along each chromosome. So it starts in dots at leptotene, and then as you progress through, these dots begin to coalesce into long strands, and those strands are the scaffold along each chromosome. And then the zipper protein is shown in green. It's a protein called SCP1, and it begins to zipper up these chromosomes in zygotene and into pachytene, and the green on red signal becomes a yellow signal. So you see how those chromosomes are now zippering up. And then by pachytene, all the chromosomes are now nicely zippered up completely along their lengths, except, and this is a mouse, I should tell you these are all mouse pictures, and this is the X and Y chromosome, and you see a little dot of yellow where the X and Y chromosomes are paired in their pseudo-autosomal regions, their regions of homology. So this process is very critical. It follows that any problems that happen early on in prophase 1 are likely to be very deleterious to the cell, and that if these if this problem persists, it's unlikely that the cells are able to keep those chromosomes together and therefore keep those chromosomes together for long enough to then separate them equally. And when this unequal division takes place, or unequal separation of chromosomes take place, this process is called non-disjunction. And we all know that that non-disjunction can give rise to eggs and sperm with the wrong number of chromosomes, and it's studies by Pat Jacobs and others that really laid the foundation for our understanding that this could truly be the causative factor in many human chromosomal conditions, XXY and Turner syndrome just being two examples, Down syndrome being another, of course. So just to complete the process of prophase 1, once those chromosomes are tethered together and recombination has begun, the protein structure, the synaptonemal complex, is no longer needed. It begins to break down, and the chromosomes repel each other because they're negatively charged. And what becomes apparent then is those DNA-DNA interactions are tightly enforced. And so by the time the cell exits prophase 1 and is ready to go into the chromosome separation phase, those DNA interactions are what are keeping those chromosomes together. So not only is synapsis and the structure of the synaptonemal complex important for keeping chromosomes together and ensuring their equal segregation, but the formation of these crossover structures or recombination structures is also critical to maintaining chromosomes together. And again, studies by a large number of groups have shown that mismanagement of these, the placement of these crossovers is absolutely critical in conditions such as Down syndrome, for example, where there are 3 copies of chromosome 21. So my lab's interest really is in these stages and how both the synaptonemal complex and these crossover structures function to result in accurate segregation of chromosomes. And the problems associated with these events in humans are manifold. This is a similar image to what I just showed you for the mouse, but this time we're looking at a human oocyte, a human egg, in which you have 23 very nicely paired chromosomes. The green stain is that stain that localizes along the chromosomes, and the blue stain is the centromere. So you see 23 centromeres, but actually you don't. You see one more than you should do. So here, this guy here is 3 copies of chromosome 21 that are desperately trying to pair and desperately trying to do what all the other chromosomes are doing. And they do, to some extent, pair, but they're obviously abnormal. We want to understand how this situation arose, but also in terms of clinical management for adults bearing 3 copies of chromosome 21, as an example. We want to understand what the impact of these 3 copies of chromosome 21 are going to do to an egg or sperm or whatever. So this chromosome could be, this trisomy could be any chromosome that you could imagine. The interesting thing is that when you look at humans as a meiotic species, we're absolutely terrible at doing meiosis. You know, meiosis is the fundamental cell division event that gives rise to the next generation, and yet we really suck at it. If you look at the lowest of our eukaryotic brethren, yeast, they're really, really good. There's a very, very stringent mechanisms, these checkpoints that Dr. Badouk referred to, very stringent mechanisms in place to ensure that all those chromosomes pair as they should and separate as they should. Even in mice, a very closely related species, they're pretty good at it too, but when we get to men and to women, we're really bad. And we really, even after 40, 50 years of research into this, we really don't understand what the underlying basis for this is. This number for women is absolutely shocking. Of course, we know maternal age has a huge part to play in this, but we really still don't understand the basic mechanisms, given that for Down syndrome, as an example, 80% of Down syndrome babies are born to young mothers. So age is not the only factor involved. So if we look at trisomies, for example, I always use Down syndrome as my basis for all my examples because it's an autosome, and as Dr. Badouk said, the problems with dealing with sex chromosomes is they are sex chromosomes and they're different to all the other chromosomes. So chromosome 21 is one of our characteristic examples, and the incidence of chromosome 21 is high, but so is the incidence of XXY. So these chromosomal segregation issues are equally occurrent in the autosomes and in the sex chromosomes. And this is illustrated here in a table from Terry Hassel's group. Terry Hassel, incidentally, was one of the offspring of Pat Jacobs' lab. So he has really carried the torch for the whole Jacobs empire. And we see that in terms of the proportion of XXY as a total number of trisomies, it's still a very high number of live births. If we look at why these trisomies occur, and I have to apologize here to any geneticists that XXY traditionally isn't a trisomy, but I'm using the word trisomy in terms of the wrong number of chromosomes. But if we look at where these events occur, the interesting thing is, well, there are 2 interesting things for XXY. First of all, when we look at paternally derived trisomies, it's equal to that of the maternal trisomies. Now, this is different to Down syndrome, for example, in which trisomy 21 is almost always a maternally derived event. So this tells you that missegregation of the sex chromosomes can happen equally in males versus females. And if anyone's interested, I can tell you some of the reasons for that. So that's an interesting feature. But if you look at maternal and paternal trisomy or XXY events in particular, you see that the majority of these events happen in meiosis 1. So I'm not going to go into why these are different, but if you look at the total percentage of trisomies arising from male versus female germ cells, I mean eggs and sperm, you'll see that it's almost all in meiosis 1. So that, again, underscores why we want to study prophase 1 as a causative factor for these type of events. So I just want to summarize some of these important features compared to the important thing about... I knew I shouldn't have pressed it twice. My slides are going crazy. Okay, so compared to trisomies of autosomes, I've said most of this, XXY occurs much more prevalently in males. And so this is a really important feature for us as scientists when we look at both male and female meiosis side by side. It's very important for us to understand why XXY in male germ cells persists, whereas other non-disjunction events don't persist. So a trisomy of chromosome 21 or 2 copies of chromosome 21 missegregating in a male, in a sperm versus an egg would kill that cell. It would not be allowed to survive, whereas missegregation of the X and Y in a spermatogonia would not kill the cell. We don't understand why that is. We have some clues. But the most important thing, as I underscored, is that as with most trisomies, most of these errors are due to prophase 1 issues. So the goals of our study, given these observations, is that we want to understand how prophase 1 events might be responsible for these conditions, and particularly how errors in recombination might be responsible. It's very important for us to understand the common mechanisms in male versus females. We're only beginning to understand now why, how different males and females are in the way they make eggs and sperm. The textbooks would say it's the same process. It really, really isn't. And studies, the genetic studies we and other groups have done really underscore that. And I've put this in bold here because it's a very important feature, to use comparative genomics to understand why these conditions are so prevalent in humans. So I can tell you yeast have an error rate of 0.001%, and women have an error rate of 30%, but unless we compare across species, there's no point in giving you those sorts of data. And that's really what prompted me to move from a medical school to a veterinary school, because I really believe that understanding these events in humans and animals can only be done by comparative approach. We're very, very fortunate to live in a genomic era now where we can study these genomes, and a new one is being sequenced daily. So we can really use the power of genetics across species to understand where we're going wrong, if you like. And then hopefully, and this is down the line, to understand and treat the consequences of chromosome imbalance in eggs and sperm as a therapeutic tool. I'll leave that sort of huge goal to people like Dr. Paduk. But anyway, so a little bit of the background of why we do our studies in mice. First of all, our mouse strains are very genetically defined. We have very, very clear information about the genome of each animal in our colony. If I went around and genotyped every one of you, you would be very, very different, and you would be a lousy control group as a start. But our mice make very good control groups. And we can use those, therefore, to understand very easily how the human genome impacts our disease capabilities. With our genetically pure mice, we can then manipulate their genomes and look at what the downstream consequences are, so we can understand what genes are involved in the processes such as meiosis. We can look at all ages, so the ability to look at developmental ages is really critical to the sorts of work we do. Female meiosis, for example, takes place in the embryo, so it's a much more tractable event to be able to do this in a mouse embryo than a human embryo. The power of comparative genetics, I've already said. And again, to underscore that mice aren't the only animal out there, and there are many other organisms that we can begin to look at that have diseases and disorders very similar to humans. Dogs, for example, have a huge number of related conditions that we can start to use as a basis for understanding human disease. So to go back to meiosis 1 now, I've done my little soapbox bit. we're looking at these critical stages of prophase 1. So remember, we have these 5 stages of prophase 1, leptotene, zygotene, pachytene, diploidine, and diakinesis. And during that time, these homologous chromosomes have to, well, first of all, they have to replicate their DNA, then they have to find each other, glue each other together, and undergo these events called recombination. And these occur at these structures called meiotic nodules. And they start very early on, but it's not until later that the true sites of recombination become apparent, and that's important to know for later. And we've looked at many genes involved in this process, but the family of genes that we focus on most are represented by these 4 circles here. These are genes that are involved in DNA remodeling, if you like. They repair DNA, and they're very much involved in many other conditions, such as cancer conditions, because if you imagine that cancer is a DNA destabilizing event, these proteins can go wrong and cause different types of cancer, but that's not the focus of my work. Instead, what we've done is defined how these proteins are involved in meiosis. And I'll just give you a little bit of data to explain the sorts of things we look at. So MSH4 and 5, sorry, I should have explained myself a bit more. These are partner proteins. MSH4 and 5 are proteins that bind together and then bind to DNA, and they recognize these early structures called meiotic nodules. They recognize these structures very early in prophase 1, and then they recruit these other 2 proteins called MLH1 and MLH3 that bind to them at these meiotic nodules. And so we know that they're important because if we look at the localization of these proteins on these chromosomes, we see them. So basically, you're just looking at red dots. Again, this is a human oocyte, a human egg, so that's at an early stage of prophase 1. And we see MSH4 localized in discrete dots all along the chromosome. So we know they're important, and we can count them in mouse eggs and sperm and human eggs and sperm and see that their number correlates with the number of recombination events that you might see. And then if we delete them in the mouse, so this is the great thing about the mouse, we can then say, well, let's take this gene out and see what happens. And this is what we've done with MSH5 in this case. So these left-hand panels are normal mice, littermates, and these right-hand panels are mutant mice. And you can see immediately that when you look at these testicular tubules from these mice, they look very different. If this was a human male, you would say that this mouse had Sertoli cell-only syndrome. They basically have no cells going through meiosis. So they have the spermatogonia, the initial cells that populate the testis before birth, and then will give rise to these waves of cells going through meiosis. But these cells don't progress further. And then you begin to see why when you look at the chromosomes. So these dots are just markers that we use for chromosome progression through prophase 1. This is that protein that forms the scaffold along the chromosomes. You see in a normal cell, you see 20 nice pairs of chromosomes in the mouse, wild-type mouse. This is the X and Y again. And you see rather artistic but very, very messed up cells in the MSH5 deficient mice. So these cells basically show that the scaffold forms, but it's never going to come together. There are few bits of chromosomes that come together, but really they never pair. And then if we look at those other 2 proteins, do you remember MLH1 and 3 that come on later, you find that they, again, form really nice dots along the chromosomes. And in fact, MLH1 and 3 are now categorized to be the ultimate final decision makers in where recombination takes place. So you see, we can define several rules. There is at least 1 per chromosome. If you think about chromosomes that have to be glued together, you need at least 1 of those sites per chromosome. And there are never more than about 2 or 3 in the humans. So MLH1 and 3 form these discrete dots on the chromosome, and that's where recombination will take place. So again, not surprisingly, when you delete these genes, either one of them, you get basically no progression. You get a little further than with MSH4 deletion because these proteins are required later. So you see cells going to pachytene, nicely paired, but they don't go any further. And if you look later on, these are cells that should be beginning to divide. A wild-type cell, normal mouse, you see chromosomes that are sort of in these very strange configurations. They're there because they're stuck with their DNA to each other and they're about to separate. Whereas in the mutant, and if you counted these, you would see 20 of these because there's 20 paired chromosomes. If you counted these, you'd see about 40, 30 to 40, because most of their recombination sites are gone. So we know these proteins are critical. We can see them at the electron microscope level. So this is one of those meiotic nodules, and it's got black dots. These are gold-labeled antibodies to the proteins that we're interested in. And we see different size dots because MLH1, MLH1 is stuck to a big gold grain, and MLH3 is stuck to a little gold grain. And so you see mixed gold grains showing that we have both proteins there. But of course, in the mutants, you either see no meiotic nodules, as in this case, or very few, and when they do have them, they have very little protein on them. So we can see that at the cytogenetic level, visually, these crossovers are completely messed up. And I'll get to what all this means in a second. Genetically, we can score this genetically as well. We can say how many times do we see maternal and paternal chromosomes mixing. So we know that all of us have mixed genes from our mother and father, and we can do the same in the mouse. We can say how many times does this genetic information transfer, and what we see in these MLH3 null mice shown in yellow and the MLH3 compared to the MLH3 normal mice in red, you see that these crossing over events are pretty much gone in the mouse. We can use this as well, and we can begin to say, well, what happens across the genome? Let's not look at one event or two events. Let's look across the genome, and this is some of the more recent fun stuff that we've been doing to really use MLH3 as a way of assessing where crossing over occurs. We still know very little about how crossing over occurs, where these events occur. But we know that MLH3 and MLH1 are defining these events. So these are the sorts of long-term goals that we can use to really assess the genome on a global scale. And again, this is showing you, millions and millions of base pairs along chromosome 17 as an example. And I've just highlighted this little spot here where we can show that if we look at this one little spot, we get MLH3 accumulation. Sorry, this should read the MLH1 accumulation, and not surprisingly, MSH4 and 5 accumulation because they're all there together. And this just confirms to us that these proteins are physically on the chromosomes where they should be and together. So what does all this mean? It means, of course, that mutations in genes such as MSH4, MSH5, MLH1, and MLH3 can all cause meiotic disruption. But the problem with this is that when we disrupt these genes so dramatically, we end up with no sperm. The mice are all infertile, as we would predict. And this doesn't really recapitulate the human condition at all. It's very unlikely you would see a complete deletion of a gene in a particular human. And so they tell us a lot about what the gene's doing, but they don't tell us much about the human clinical condition. And so they don't really recapitulate the disorder. But what we know and what the genome projects have told us in the past 5 years is that there are variations among us. I said that if we genotyped all of us, we'd be very, very different. And we're different because of these subtle variations in our genome, subtle single alleles or single base pairs that are mutated. And these are called SNPs for single nucleotide polymorphisms. And there are millions of them in the genome. And what we don't know is which of these SNPs are likely to cause a disorder and which aren't. So which SNPs are likely to make me responsive to a drug and Darius not responsive to that drug. It could be as simple as that. Which SNP is likely to give me diabetes and Darius not. There are so many different events that you could imagine these SNPs could regulate in very subtle ways that may never be affected until we're exposed to some toxin or some environmental event. And so understanding what genes does is just the start of what we do. We need to now take this into the translational setting and look at subtle variations and how subtle variations may predispose to various disorders, for example. So I'm going to skip this. What we tried to do, and the last point I made on that slide, was that we need to repeat this, of course, in human work. And I'm just showing this to remind myself to tell you that as I did before, that female meiosis takes place in the embryo. What we've done is we've started to look at human meiosis and the errors that are around in human meiosis by analyzing human oocytes from patients who have donated fetal tissue to us. And so what we can do is we can take fetal ovaries and begin to look for sort of molecular markers of recombination and see if they're there. No one had done this until about 6 years ago. And so I'm not going to bore you with all the data, but we can take these human ovaries and we can stain them for any of our favorite proteins and see if they're there and see if they're there in the right place and at the right time. These are just some examples, and we can create these very long and complicated graphs showing you where they go up and where they go down. But the take-home message from this is that the events that are taking place during prophase 1 in humans are exactly the same as in the mouse. So the mouse is a very good model for the human in this respect, except in one instance. And that is when you look at later stages of prophase 1 and you look at my favorite proteins, MLH1 and MLH3, and you see the variation in how these proteins accumulate on the chromosomes is really rather startling in humans. So what I'm showing you here are basically, we've counted MLH1 dots on chromosomes. And so this red bar here on the left shows you all the dot counts from single oocytes, single eggs. And you see the variation goes from something like 10 to 110. It's a huge variation, tenfold variation. Now remember, there are 23 chromosomes, pairs of chromosomes in the human genome. We need one crossover at least for each chromosome. 10 is just not going to cut it. And similarly, if you look at 110, you think about how many dots along those chromosomes there are. The likelihood that that thing's ever going to come apart when it should do is really unlikely because it's glued in so many spots. So we can automatically say, well, we would predict that these larger numbers are going to be potential risks for chromosome segregation. And similarly, these lower ones are also going to be potential risks. So we automatically know that compared to mice and yeast and zebrafish and almost any other organism you look at, humans have this enormous variation. Remember, this is in the embryo, so this is not a maternal age effect. This has already happened. And then we can look at single ovaries. So each of these yellow columns here are the counts from single ovaries. And we can speculate, perhaps, that in individuals where they have particularly low counts in every egg, maybe those ones would be potentially at higher risk of chromosome abnormalities and chromosome segregation abnormalities. So this is all very speculative, but it's the sorts of things that we want to address at the clinical level.
Can I ask a question?
Yep.
We go back to this slide. So you're saying that if you have an elevated MLH. So, am I understanding this correctly? You would anticipate it if somebody has a very high number, let's say on the X chromosome of the MLH1 focus, it's associated with non-disjunction.
We can't make that link. So
you cannot make this link
this
yet?
MLH1 and MLH3 is here in the embryo. So these are embryonic ovaries, embryonic eggs. It's going to be 30 years before those guys segregate their chromosomes. That's why.
Have anybody looked at the Klinefelter, in the testicular tissue or any?
None of this has been done.
Okay.
Yeah.
So see?
Yeah. No, I know. It's hard to get these specimens, and it's only been since this paper came out. This is our work in 2005. Really, no one really even expected this. No one predicted it. So it was quite a shock at the time that there was this huge amount of variation. I mean, when I tell you that if you look at 1,000 mouse eggs, almost every one of them will have 26 MLH1 dots. Almost every one of them. I mean, the variation is so subtle. And here you have a tenfold variation. I mean, it's just unexpected. Everyone said it's maternal age, so it doesn't matter what happens in the embryo, it's later that the problem's going to arise. And there's no doubt that the huge component of it is, I'm not saying this is the only problem. remember that there are many what we call the bad eggs here, but 6 million of these eggs will die at birth. Right? So, each woman is born with 1 million eggs that came from a pool of 7 million. So we don't even know whether this lower half are just cut out. It's not known. So what we can begin to do is look at high and low numbers of MLH1 dots and correlate them with SNPs, variations in genes that we think are important. And this is going to be a very long-winded and laborious task for some poor person in my lab. Anyway, so that's one of the questions we want to ask. Do SNP alterations play a role in meiotic dysfunction? Do environmental factors, so every patient that we obtain tissue from, we ask a series of questions related to their environmental exposure. So again, another scary topic. It's not what you do to your body, maybe, it's maybe what your grandmother did to her body, and how your eggs developed in utero. So it's a very cross-generational effect that we may be looking at in terms of maternal meiosis defects. And then one of the big problems we've been having with human female tissue is that we can only get ovarian tissue up to 24 weeks of gestation. So that's the abortion limit. And so what we did is we took out a leaf out of Dr. Travis's lab and book, and we said, well, can we culture these tissues in some way? People have tried for years. In fact, here at Weill, Cornell, they tried for years to culture these tissues from postnatal ovaries and weren't able to culture them very far. And so what we did was we took fetal ovaries, human fetal ovaries, and we put them into immunocompromised mice and cultured them for various periods of time in a xenoculture experiment. And so what I'm showing you here is an ovary from a 13-week-old fetus in section. Human fetus, sorry. And it's packed full of eggs. I mean, this was an amazing thing to see for the first time. Chock full of eggs, and they're all in prophase 1 or early prophase 1. When you culture these human ovaries in a mouse for 12 weeks, you begin to see startling morphological differences. So these are the eggs here all going through prophase 1, and suddenly you have whacking great big follicles. So these eggs are progressing through prophase 1 normally. Within 12 weeks, we get development that we would expect to see in 20 or 30 weeks of development. So really, this is an exciting way for us to proceed and to look further as to what the developmental consequence of these events are. And just to show you one of our trial experiments, in this case, we took a 23-week-old ovary. We put a piece of the ovary under the skin. So we put the tissue under the skin or under the kidney capsule, two very, very routine methods. And then we culture them for various periods of time. And in this case, we cultured for various periods of time and then gave 10 weeks of FSH treatment, which is sort of to recapitulate the adult human environment. And what we find is under the skin of a mouse, we suddenly have a tissue that looks like a human ovary. So we have, this is a huge follicle with an egg inside. There's a higher image here. And this egg looks, to all intents and purposes, absolutely normal. We're not going to try any Frankensteinian fertilization experiments or anything here, and we're not trying to do anything scary. We're trying to say, can we follow these eggs further and see what their developmental potential is like after aberrant chromosomal events. And we think that with this model, we can start to do that. So in the future, for these xenotransplantation experiments, of course, we want to assess the progression of these oocytes through. That was one of the very first experiments we did. We've done a load more since, and we have some very good clues as to what we can begin to look at in terms of meiosis, but also about follicular genesis in terms of situations like Turner syndrome, where the ovary is completely destroyed because of this, because of the meiotic program. We believe that we can begin to look at how the eggs interact and communicate with the cells around it. We can begin to explore the impact of environmental toxins. Bisphenol A is one of these scary toxins that's in everything we're drinking and everywhere we are, and it's known to have a huge impact on progression of meiosis in mice. It's not unlikely that it has the same impact on human meiosis, and we can begin to explore that. And of course, we want to do the same analysis in sperm. And some other approaches we're taking is to look at primates. I mean, primates really have been very underutilized for these sorts of studies. And one of the first questions I asked was, well, if humans get birth defects, birth defects and chromosomal abnormalities, do primates? No one's ever looked. It turns out there's no data out there. And so we just started a collaboration with the Oregon Primate Center, and it turns out that if you look at primate monkey eggs, their chromosomes are all over the place. They look like some starry night. They're all over the place. And we're just beginning to explore that, and I think that this is a really, really good way to utilize the primate model. We would like to team up with people like Darius to screen patients for different variations in different genes to really try and get a handle on what how different SNPs might impact human health in any number of disorders that you might imagine. And then to take those SNPs back into the mouse and say, well, if we make that same variation, subtle variation, can we recapitulate the phenomenon that we're seeing in the human? And doing things like developing live imaging tools to be able to look at chromosomes and the way chromosomes interact is something that we're very big on at the moment. I'll just show you a few pictures of some mouse eggs. These are live mouse eggs. They don't look quite as nice as those beautifully stained ones because these are a pain to get. But we can begin to look at how chromosomes interact and get together. And again, this is one where the chromosomes are just beginning to find each other. They form this lovely structure called the bouquet where they come together and try and find their partner. And then this is another one. Again, the chromosomes have found each other or almost found each other. And in fact, if I zoom one of these guys up, you can see that this chromosome's all paired and then it bubbles out and it's just going to zipper up completely. Remember, these aren't fixed chromosomes. These are live ones now. So the potential for what we can do with these is unlimited. I'm not going to bother with the conclusions. Oh, no, apparently I am. Okay, I'm just going to thank the people involved. This is my lab, past and present. These three individuals, Nadine Kolas, Anton Svetlanov, and Kim Holloway, have been really instrumental in the analysis of MLH1 and 3 in the mouse. Michelle Lindsay was the super student who initiated all the human work. Really, no one had ever done much of this before. And Rikan did all those beautiful xenotransplantation experiments and really pioneered that technique in my lab. Shanfei Sun and Meisha Morelli have been working on the live imaging and some of the downstream MLH3 effects. And of course, our funding source of the National Institutes of Health, the March of Dimes, and the Hereditary Diseases Foundation. And then I'm just going to give a little plug for the Cornell Center for Reproductive Genomics. This is really a new center, very, very new, that's stemmed out of people like Darius and Dr. Schlegel and others who have been so excited to get together with the veterinarians and the veterinary scientists at the veterinary college to bring together people with common interests in reproduction and genetic mechanisms. And I think that this sort of endeavor and this partnership is really what makes the Cornell institution a very strong place to do research. So thank you, and I'll take questions.
Thank you. You know what? I have a question. Do you think that this, your ovarian model, actually is going to help us to understand triple X and Turner syndrome? Because it seems like a natural...
Yeah, I mean, really, I was shocked that nobody had really looked at why the ovary degenerates in Turner syndrome. I mean, you know, there's this crosstalk between the egg and and the stroma around it, but no one really knows what that crosstalk is. And it really only became apparent to me when we started looking at these mutant mice. So I showed you that in some of the mutant mice you get no sperm. I didn't show you the ovaries of those mice. When you kill meiosis early, like early in prophase 1, you get a mouse with no ovary. And in my first paper on this, I suggested this was a model for Turner syndrome, and the reviewers made me take that comment out. But I really believe that it was the first indication to us that there's some crosstalk there following the end of prophase 1, or following when the eggs go into arrest. I think these models are there, and we could look at them. I think it would be great. And we do occasionally get tissues that are XO, and we can look at, but we get one every now and then. It's not a very powerful study with the occasional sample, but we can look at it. Yeah.
Well, probably a lot of people don't realize how difficult it is to actually establish models like that, and especially from the embryo. It's just not that simple, taking the piece of tissue and putting an ovary into the mice. So that's a pretty amazing work. But you know what? From all this kind of a bouquet of early meiosis 1 genes, right, and the proteins which you look, which one do you think would be the focus of looking in let's say Klinefelter syndrome? If we want to define, which the genes are the targets for then genetic screening, because we've looked at MLH1, we haven't looked at Klinefelter syndrome. In a Klinefelter man, we've looked at the azoospermic man, but we've looked at that, and MLH3 already we screened. Which one did?
MLH1 and 3 are too late. They're too. I think that the decision is made already, and if Klinefelter and Down syndrome and any of those single chromosome abnormalities are not like the mice we're seeing, where every chromosome's falling apart. I think that they're much more subtle, and they're something to do with the decision. I said that management of where those crossovers go is critical. You need one per chromosome. You don't need them too spaced apart. You don't want them too spaced close together. I think that management issue is where it's going wrong, and we see that in the human eggs all the time. And I think that if we can understand what genes are selecting, so you saw the MSH4 and 5 dots, there's hundreds of them, right? And from those hundreds, like in the mouse, you have 350 MSH4 dots. And from those 350, MLH1 and 3 select 26. Which 26 do they choose? Do they pick every other one? Do they pick every other two? Do they go yes, 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 no, and do it randomly? We don't know, and I think that is the gene that you have to pick on. And it could be MSH4 and 5. It could be a gene called MUS81, which we're working on. There's a whole slew of them. It's not likely to be one gene, I can promise you that.
So you think that it needs to be before the MLH1?
It's way before.
It's not going to be just a one.
Some people actually tell us it's before MSH4 and 5, and it's before chromosomes even pair, so I don't believe that. But I think it's before that.
So if you plan for the future, we would pretty much look at those genes which are before, and then looking at, at whether there are single nucleotide polymorphism in those particular genes in the men who have Klinefelter syndrome versus men who don't Klinefelter syndrome. That would be the strategy.
I would say that what we would love to do in an ideal world is screen infertility patients for all of those genes, find the SNPs that they have, and say, well, are those SNPs, are those the ones that are causing their subfertility? Put those SNPs in the mouse, make a mouse with, instead of the complete deletion of the gene, just change a base pair and just twiddle and tweak and see whether we get subfertility. And that's the way to do it, not the way we've done it with the huge deletions. I mean, we know what the genes do, that's good, but now we have to do it, be more subtle.
But looking at your experience, this is almost like, 30 genes? How many of those genes are like in the mice and homologs?
It depends who stands up here as to who will tell you. I would tell you my favorite genes. So, my favorite genes are the most important ones.
So,
how can I tell you?
See, because I was looking at Lenny, how she fascinated look at the ovary, because obviously you created a beautiful model for a lot of the studies which she's very interested. But kind of looking at the future, it took for MLH1 and MLH3, which was screening, it was almost a year of work, because as you know, those genes are extremely complex. So I'm kind of thinking for, how much work it would require, if you think that there are probably 10, 15 genes which are probably critical to screen. That's a pretty dramatic amount of work.
If we
But
get our funding for the center, I'll screen every gene you want me to screen. How about that?
let's hope for that. So I will. Well, thank you very much. Any other questions?
This is a kind of a sensitive topic, but the number of abortions for people who have pregnancies of XXY, XYY, the triple X is very, very high.
Yeah.
It's been a sadness to all of us, but I've often wondered, could that conceptus be helpful to you?
Absolutely, and so we have, you know, Darius has mentioned the IRB hell that we all go through.
Right.
Part of the way we obtain our tissue is the patients are not, we're not allowed to discuss research with them until they've agreed to the termination, and nor would we want to.
Right.
The time between when they sign the consent for the termination and the termination itself is quite short because they've already had a few days to think about it, and it's a highly emotional situation. And so the time there to discuss these issues is very limited. Some patients come in saying, I want to do research, I want to be involved, but very few do because they've got way too many other issues on their plate. So we have a very limited time frame, and therefore the ones who are going through a more emotional and a more emotionally charged termination, such as those with any kind of condition, are less likely to donate. We have had some. You know, only 50% of them are female. So again, we can only look at female tissue at that stage. Certainly, I hope that we'll be working on...
Only female tissue?
We can only look at female tissue because we're looking at the eggs, remember, going through prophase 1. So we would need, you know, postnatal male tissue. And hopefully with Darius, we'll do some of this stuff now. I mean, we're just building up to it, and hopefully we can begin to see if the same phenomenon apply. For example, all that variation in MLH1 that I showed you in the oocytes, no one knows if that really... Well, people have done some studies in the male, but not that many. So we need to address both the male and female together, and it's our access to tissue that's limiting us.
Okay, thanks.